thank you also to the audience for being here. I'm glad, I'm very happy of presenting this topic about our research in the last years in our clinic in Valencia. The learning objectives of my talk would be first to describe the upper limits of assisted reproduction and the currently reached efficiency, discuss also about the relevance of establishing valid and robust techniques to determine gametes quality, also describing gamete quality parameters and their relevance in embryo development and pregnancy outcome, and finally to define how these gamete quality measurements can influence about the RT outcome. As I show you in this slide, when we try to represent the upper limits of assisted reproduction techniques, even from the best sperm viewpoint, meaning that we are using donated sperm, you can see that the cumulative live birth rates achieved even using own or donated oocytes are far from being perfect. In this sense, not every patient succeeds, and there are several attempts needed in some cases. The ideal situation will be having from each oocyte and each sperm employed one embryo that finally will result in a newborn. What do we mean with quality when referred to gametes or embryos? A quality indicator should be an intrinsic feature or trait linked to optimal results under a functional viewpoint. In our case, the optimal result is obviously having a healthy newborn at home. Let me raise this kind of poll in the audience. Okay, what do you think once you have the oocytes, the sperm cells, or embryos in the laboratory is the most important, the gametes, the embryo, which gamete, concerning the assisted reproduction results? I guess that given that mainly the audience is composed by gynecologists or even embryologists, and I am not an embryologist, some of you would say, okay, identifying the best embryo is the most relevant issue in assisted reproduction treatments, because you can then forecast about the live birth probabilities, you can select the optimal to get the pregnancies earlier, but we must assume that this is only the case if the number of embryos available is higher than the number of embryos that are going to be transferred, and it's only the case when you have a cohort of embryos with different qualities. Okay, it seems it's not so important. Moreover, after the vitrification techniques have been available, its interest is slightly decreased, because if you are not choosing the right embryo, then it will survive freezing thawing, and then you will achieve live birth in the next cycle. That said, you can also say, okay, the oocyte is the main actor in this picture, but by identifying the best oocytes, you can forecast live birth probabilities, but you can't choose the oocytes. Even thinking only in countries where a legal limit of inseminating oocyte is established, you are going to use all the oocytes you are going to you are having in the laboratory. Then, in my opinion, and maybe against 95% of all of you, I think that identifying the best sperm should be the situation where you are improving the results in the assisted reproduction laboratory. Why are sperm cells interesting? They are almost always in excess. As I told you, it's not a limiting factor. It seems to be the oocyte. You always have more sperm than oocytes. Moreover, they are genetically unique, because each sperm is in an individual issue, and they are presenting several features that can be linked to success or failure in assisted reproduction. In fact, there's a recently published paper showing that within the same male, once you perform the complete DNA analysis of each sperm, they are very different among them. This is all explained because of the spermatogenesis process, as you may all know, and there are two key steps. One, the first, when the chromosomes are separated during the process, and second one, on the recombination. And this will mean that within an ejaculate, you may find good, bad sperm, and you may only need a few, so we need to choose wisely. Then, once you are in the andrology laboratory and the IVF laboratory in your clinic, you face this kind of situation where you have the different steps in the assisted reproduction procedure, and we will only focus first on the sperm analysis, and second on the sperm selection, trying to forecast the embryo quality, and finally the involvement in the reproductive results. We find several limitations when analyzing this data. Interestingly, in human reproduction, you find that when you try to study infertility on an individual, you always find a bias which is included by the partner. In this sense, several studies are not taking into account the other side of the picture, and this is confusing frequently the results obtained. Regarding sperm quality markers, you may find either morphological, biochemical, or even molecular, and in the last ones is the one we are going to focus our talk. You all know that the only tool that we are usually employing in the assisted reproduction laboratory is to forecast the male fertility or a sample fertility is the World Health Organization-based sperm analysis. In this sense, I think we agree that the forecast, the predictive value of this analysis to forecast live birth is limited, that you may face this situation frequently when you have normal results in infertile males and inversely abnormal results in fertile males. When we employ this data to forecast fertility by means of our ovum donation program in Valencia, you may detect here in the Broad curve analysis that neither the raw sperm volume, sperm density, the volume of the ejaculate, motility, and even morphology, none of them are good predictors of the consecution of a live birth. Regarding the molecular profiles that have been today described as sperm quality markers, it seems that's very difficult being a successful sperm, because you need to be healthy in several aspects. Those linked to nucleic acids, such as aneuploidies, DNA fragmentation, correct gene expression, epigenetic, and those non-linked to nucleic acids, such as the apoptotic events, oxidative stress, hyaluronic acid situation, ubiquitin, and so on. Regarding one of the most studied, regarding DNA fragmentation, I would like you to summarize and present some of the facts. First of all, when you see the data and you analyze all the papers that have been published to date, you may find an agreement, which is that there are very highly controversial results. Moreover, this may be caused because different techniques are employed in each study. There are different cutoff values and odds ratio of achieving a pregnancy, and outcomes are not always related to miscarriage or live birth. I would like to present some of our own results that we have been doing these years. And in IVF and ICSI, we found several embryonic traits that have been related to increased DNA fragmentation, such as fertilization rate, worse pronuclear pattern linked with samples showing high DNA fragmentation, more asymmetry in embryos coming from these higher sperm DNA fragmentation samples. But the conclusions to date seem to be included in these three papers. First, there's a mild correlation between the sperm DNA integrity and embryo quality that could potentially affect success. That seems to be true. Nevertheless, the clinical relevance is at least moderate or low. The prognostic value is very low, and it seems unnecessary to complement each of the basic sperm analysis. This information may be helpful after repeated treatments with bad embryo quality, including a normal ovarian response and other normal sperm parameters. There's a link that has been described between pregnancy losses and DNA fragmentation in a very recent meta-analysis from Dr. Sini, which I think it's the best information available in this sense, but obviously further information is needed before we can offer this to every patient. Regarding oxidative stress in sperm, you may all know that oxidative stress is a physiological, almost physiological situation that sperm samples may face, and this will be finally confirmed because of the balance or imbalance between the reactive oxygen species and also the antioxidant capacity of the sperm samples. Several papers have been published relating this with infertility, with male infertility, and also with embryo quality. I will bring you some of them here. This was a paper we published several years ago where we linked the female factor standardized by the means of the ovum donation program with the expression of some genes that are related with the protective capacity of sperm against ROS injury, showing that there's a correlation between embryo symmetry and also the percentage of optimum blastocysts depending on this antioxidant capacity in the sperm cells. And in other order of things, I brought a couple of slides regarding IMSI. This is not a clear molecular marker, but some studies have linked the percentage of DNA fragmentation with the number of vacuoles that have been detected by means of this technique. And I brought this because first of the link with DNA fragmentation, and second because there are randomized control trials that have been already published. In fact, there's also a meta-analysis pointing to an improvement by using these techniques, and this may be achieved supposedly because of the avoidance of DNA fragmented sperm once you inject the sample. I think you all know which are the methods that are employed by these techniques, and several reports have demonstrated increased implantation or pregnancy or live birth rates together with a significant decrease in miscarriage rates, mainly on some selected patients' populations, meaning couples having repeated ICSI failures and also patients having very abnormal sperm parameters. This is the last one I want to show. Then, regarding IMSI and the clinical aspects, seems that there are good scientific evidences, clinical related to miscarriage, on selected patients' population. One of the pitfalls should be considered, which is the cost of the equipment, the embryologist training. It's time consuming, but this is another option. Other molecular marker that could be employed even to select sperm is the ability of sperm cells to bind hyaluronic acid. This is based on the fact that diminished sperm fertility is associated with the retention of surplus cytoplasm, and this means that immature cells fail to undergo this membrane remodeling, thus they are not able to bind the hyaluronic acid bound plates. Then, apart from being a sperm quality indicator, these sperm cells can be selected for ICSI. There are a couple of studies available in the literature, mainly from Italian groups, and some of the studies have been showing an improved sperm quality among those that have been bound to the plates, but the clinical relevance to date seems not to be very important. In the second part of my talk, I would like to talk to you about the last 5 years, 6 years almost research about the omics in sperm. We feel that the best option, once you have the tools, is studying a lot of things at the same time, because only studying DNA fragmentation, only studying oxidative stress, only studying other molecular aspects are only providing you with a partial view of the whole picture. In this sense, given that paternal contribution is not only 1/2 of the embryo's genomes, because there are exclusive mRNAs, proteins, and structures delivered into the oocytes at the time of fertilization, it's very important until the embryo genomic activation. And even thinking that sperm cells have no transcriptional activity, they still have a pool of mRNAs that have been synthesized during spermatogenesis, and they are going to take a role in early embryo development. In this sense, we raised the sperm fertility array based on the hypothesis that sperm cells with or without reproductive success will behave different, will present different transcriptomes. Moreover, these differences among transcriptomes will be, again, different depending on the assisted reproduction technique employed. If they were IVF, ICSI, or IOI. Then the objective was to develop a predictive tool based on microarray analysis for each technique and describe the molecular factors involved in fertility and infertility. It's like having the combination, the right combination in the K-log of sperm fertility. This is the rationale beyond this kind of experimental design. Obviously, if all these are the procedures sperm needs to be over trying to be successful, obviously, depending on the assisted reproduction techniques, you are helping from the outer side to this sperm cell to be successful. The scientific basis, it's quite easy to understand. Once you have different biological situations, in our case, samples from which a pregnancy was achieved compared with samples from which pregnancy was not achieved, you can stain them. There are several methodologies that can be employed in this sense. And once you hybridize and scan, you can have this kind of pictures about the whole genome, gene expression in these samples. Then the results they provide are a list of genes, meaning those genes that have been found to be differentially expressed, genes that are present or absent in one of the groups, and you can also perform functional analysis regarding the percentage of genes that within a biological process, a cellular component, a molecular function, or a metabolic pathway has been significantly altered. Then we published about 5 papers in this issue, mainly in fertility, sterility, and RBM online. If someone of you want, I can provide you with them. But what we get is what I'm showing you. We have the list of genes that have been found to be differentially expressed on these patients achieving a pregnancy, and also in these patients not achieving a pregnancy. And also, we have the list of genes, and here I only show the 10 mostly differentiate, that were found to be overexpressed in non-pregnant women or overexpressed in pregnant women. The same situation was established for in vitro fertilization samples, and also for those samples that obtain differential results after being employed in ICSI. Regarding the gene ontology analysis results, I show you a couple of slides because the main point of the gene microarray analysis is the main difficulty, meaning that you have a lot of results that it's very difficult to compound and to understand. In this sense, I show you the top 10 biological processes, cellular components, and molecular functions, some of them related with immune system features, with membrane, with acrosome. Also, those genes that have been exclusively found on sperm from pregnant couples, meaning that they are potential fertility markers. On the other side, those that have been exclusively found on sperm from non-pregnant couples, those that can be considered as infertility markers. And several others that have been found to be differentially expressed on genes. In this sense, we have the whole picture showing which genes have been involved in success or failure in the different assisted reproduction techniques. And we were also able to confirm that the more invasive assisted reproduction technique, the less request for the sperm to be successful. Some of these genes have been already linked with reproductive success in other tissues, or even sperm, but the vast majority of them were related with fertility with the first time. Once we define the ideal situation, then we must ask ourselves, how can we use this information to select the appropriate sperm cells for assisted reproduction? Because it's very difficult in a wet sample to decide which one is the best. In this sense, you know all the currently employed methods in the assisted reproduction laboratory, some of them that have been tested or are being tested in different settings that are not yet available in all the laboratories. But I will bring you here a couple of ideas about how, once you know which are the molecular traits of successful sperm, how you can affect the results. One of these is magnetic activated cell sorting. As you all know, magnetic activated cell sorting enables the possibility of selecting some sperm based on membrane characteristics.
You've got 1 minute only.
It's 5 minutes.
It was a mistake.
Okay.
It came from 25, but it's 20 minutes talk and 5 minutes discussion. If you can reach the conclusion. Thanks.
Yeah, I can. I have 10 more slides, no problem. I can resume in the last minute, which is once we know the key-log combination of the genes that have been expressed, all those genes that are coding for membrane-related genes are meaning that are expressing proteins that are located on the plasma membrane. In this sense, we can employ MAX technology, which has been demonstrated to be very useful regarding apoptotic sperm cells, to perform a customized sperm selection based on the molecular features exhibited by a single infertile patient. So in conclusion, to date, we have several molecular markers that have been either related with fertility or infertility. We can use a lot of them with the help of microarray analysis, and we can also use them or think about using them trying to select the most convenient sperm, trying to improve assisted reproduction results. And thank you all for your attention.